So now let's talk a little bit about nucleic acid chemistry. Now, in a way, nucleic acid chemistry is simpler than that of protein chemistry because nucleic acids can only consist of those four bases. Nucleic acids only are found in a limited number of structures, especially DNA. So the chemistry of DNA is a little bit simpler than that of RNA, but there are some important things we need to mention. First of all, again, DNA especially is typically found in its double helical B form in the cell. And even if you purify a DNA sample from a tissue, it will stay in the double helical form. Remember that the two strands are only linked by hydrogen bonds, but the hydrogen bonds are very stable. And if you have two strands that are hundreds or thousands or millions of base pairs long, then that means there are many hydrogen bonds holding those two strands together. So even though they're hydrogen bonds, which means they're not as strong as covalent bonds, because there's so many of them, it forms a very stable arrangement. However, you can separate the two strands of a double helix. The easiest way is through heating. So if you heat a DNA double helix to almost 100 degrees Celsius, almost boiling, then the two strands will separate. And when that happens, we call that denaturation. In fact, it's the same word we use to describe the unfolding of a protein because it is analogous. The same thing would happen to a protein if you heat it up and up, it will unfold. But when we are talking about DNA, denaturation means the separation of the two strands. And then if you cool the two strands enough, then they will reattach to each other, zip back together, again, according to their complementary nature. We call that process annealing. And of course, if you heat two strands enough, you can completely separate them. The more you heat them, the more they will separate. And again, this is simply due to the physical nature of the double helix and is not dependent on the nucleotides, except that there is a difference in, or you can tell something about the sequence by how hot the DNA needs to get before they completely separate. So the temperature at which a given strand completely denatures, when it's about 50% denatured, we call the melting temperature or TM. So the TM of any DNA strand is the temperature at which it becomes 50% denatured. And you can usually measure the denaturation of DNA using spectroscopy. So single-stranded DNA absorbs light at a slightly different frequency than that of double-stranded DNA. So when a sample is about 50% denatured, we say that it is at its melting temperature. And so the melting temperature is dependent on several factors. One is the length. So the longer a double helix is, the higher the temperature you'll need to get to melt it. Another thing, though, that affects melting temperature is the content of the bases. So it turns out that DNA sequences with a large percentage of GC base pairs will have a higher melting temperature than those with more AT base pairs. And so that's shown here. So the difference can be, so going from, say, 20% GC to 60% GC gives you a change in melting temperature from about 80 degrees Celsius to almost 100 degrees Celsius. So why would the nature of the bases affect the melting temperature? The answer to that question is, if you remember the way that the bases pair up, remember that an AT base pair consists of two hydrogen bonds, whereas a GC base pair consists of three hydrogen bonds, which means a GC base pair is 50% stronger than an AT base pair. So the more GC base pairs you have, the stronger the double helix structure becomes. And so the higher the melting temperature. So in fact, you can use melting temperature to measure the GC content of a DNA double helix. Otherwise, the sequence of the bases doesn't affect the melting temperature. So in fact, this just tells you the percentage of the sample that's GC. Now, of course, if let's say you get a melting temperature of let's say 85, so that tells you that the GC content must be close to 40. So let's say it's like 38%. And if the GC content is 38%, the AT content must be 62%. But that's all it tells you. It cannot tell you anything about the sequence. In other words, what order those bases are in. Now, another thing that's important about nucleic acid chemistry is that the nucleotides can be modified. And those modifications are important because many times these modifications happen to DNA as a result of either toxic chemicals from the environment or other factors that change the nature of the nucleotide, which can then lead to mutation. So a mutation occurs when you have a base in a DNA sequence that is substituted for a different base, and then that substitution eventually gets copied into the parent or the daughter strands of that sequence when it gets copied, and then that then encodes or allows that DNA sequence to either encode for a different protein or otherwise affects how that gene, wherever the mutation occurred, to function. And so there are lots of ways in which nucleotides can be transformed that can lead to mutations. One is deamination, which basically just means the loss of an amino group, usually replaced with a carbonyl oxygen. So cytosine, for example, can be deaminated into uracil. So if the amino group is removed from cytosine, it becomes uracil. Methylcytosine, also methylated cytosine, if it gets deaminated, it becomes thymine, because of course thymine is just methylated uracil. And then the purines also can be deaminated. So deaminated adenine we call hypoxanthine, deaminated guanine we call xanthine. But all of these can cause mutations because all of them have different base pairing properties than their originals. And there are lots of things that can cause deamination. One common cause are these nitrous acid precursors. So things like sodium nitrate, nitrite, nitrosamines. These are all compounds that can lead to or increase the rate of deamination of DNA. So these are can all be considered mutagens, compounds that in high concentrations can trigger deamination and therefore mutations. Now it's important to point out that it's not the deamination itself that is the mutation. So for example, let's say you had a sequence like this. So let's just say A, G, C, C, T, A, T, C. And so we'll say that's the 5' end and that's the 3' end. And then the complementary sequence to that would look like this. It would be T, C, G, G, A, T, A, G. And so this is the 3' end of that sequence and the 5' end of this one. So if you had, let's say, a deamination occurring on, well, let's say this cytosine right here, that would mean that after deamination, the sequence would look like this. You would have a 5', 3'. So everything would be the same. AG, remember that deaminated cytosine is basically just a uracil. So you would have actually a uracil where that cytosine used to be. C, T, and then the rest of the DNA would be the same. Now, the other strand would look like this. You'd have T, C, this G would still be there, G, A, T, A, G, but now you have this cytosine, or this uracil, where it's not supposed to be. So this uracil is here as the deoxy form of uracil, which again normally doesn't occur in the cell. And not only that, but U and G don't base pair with each other. So this would form temporarily this sort of inappropriate structure. But the big problem would happen if this strand gets copied, because if this DNA sequence then gets copied, each strand here would act as a template. Now, one strand would be fine. So the one on the right, which didn't, or in which the deamination didn't occur, would look the same. Make the daughter strands a different color, and so we'll call it blue. So this one would be the same, but then or have the complementary sequence. So again, if this was the parent strand, T, C, G, A, T, A, G, then the other strand would again just be A, G, C, C, T, A, T, C, and then but then in the opposite strand, you would have now your deaminated strand would still be there, so it would be A, G, U, C, T, A, T, C. But then the other strand now would have almost the same sequence, so it would be T, C. But when the DNA polymerase, which is going to be making these new strands, when it gets to that U, it's going to do, again, it's going to put a base opposite that uracil, which will base pair with the U. And the only thing that base pairs with the U properly is an A. And so that's what will get put in that spot. And then in the next generation, this will get copied again, and you'll have another copy, which is now going to look like this: T, C, A, G, A, T, A, G, and the opposite strand would be A, G, T, C, T, A, T, C. But the important thing is that now at this position, so we started with a CG base pair at this position, and now at that same spot, there's a TA base pair. And so that is the mutation. So that's just an example of how this depurination event can lead to a mutation. And so if that base corresponds to say an amino acid sequence or a regulatory sequence or whatever, that can cause the individual that inherits that mutation can now have a different gene sequence. Another type of nucleotide transformation that's important is called depurination. So remember, purines are just the adenines and guanines, and so there are various reactions that can essentially break off the purine from the ribose, and so you can end up with this apurinic residue, which is just now the nucleotide without that purine there. So there's just no base at all. And again, this can lead to mutations because if the depurinate or the apurinic residue is present when the strand is being copied, then sometimes just a random base will get added across from that now empty spot, and it will have the same effect as there being a missing base there, or having a mutation there. Another kind of common nucleotide transformation is UV dimerization of of adjacent thymines. So if you have two thymines that are next to each other in the same strand, so here we have two thymines sitting right on top of each other. Remember that in bDNA, the bases are more or less perpendicular to the long axis of the helix, so they're just sort of stacked on top of each other. If this structure is hit with UV light has the ability to cause these two pairs of double bonds to connect to each other and form what's called a cyclobutane dimer, which means that now we have a 4-carbon ring between these two adjacent bases. There's actually two things that can happen in the cyclobutane dimer, or you can have this thing called the 6,4 photoproduct. But UV light can cause either one if you have two thymines next to each other. And that causes obvious problems because the cyclobutane dimer or the 6,4 photo byproduct creates a kink in the double helix. And so that creates lots of issues. One, it prevents replication, which can cause the cell. It also prevents transcription through this region. So best case scenario, the cell will die. Worst case scenario, it will create a mutation, which can cause bad things to happen. An example of what can happen if you have a mutation is you can have, if this mutation just happens to be in part of the genome that has genes that control cell division, and there are lots of genes that control cell division. Cell division is a very tightly controlled process in most cells. Then you could have a mutation that could lead or prevent the cell from replicating properly and at the right time, or lose control of the replication process, which is essentially what cancer is. So a cancerous cell is one in which the genes that control replication don't function properly, and the cell just replicates out of control. This is why UV light, for example, causes skin cancer. So UV light from the sun can create these photodimers in DNA, in skin cells, and if those mutations happen to hit a gene that controls cell division, then that cell can become cancerous. And so all of these substances that cause nucleotide transformations, that's another thing they can do. They can cause cancer. Another really common form of nucleotide damage is what are called reactive oxygen species. So these reactive oxygen species, like hydrogen peroxide, or various oxygen radicals, like superoxide radical or hydroxyl radical. You probably know from chemistry, a radical is something that has an unpaired electron. So a hydroxyl radical has one extra electron that's not paired with another one, and superoxide is a molecule of oxygen with an extra electron. But these can form just in the process of the sort of day-to-day reactions in the cell. Usually they're highly reactive, so they usually react with other substances that are nearby. But if enough of them are present and they stick around long enough, they can react with important molecules like proteins and lipids. Typically, if they build up enough, the cell will just die. But again, they can react with DNA and create all kinds of DNA damage. And that can also lead to things like cancer or just a general wear and tear of tissue. So all of these nucleotide transformations are again associated with mutations. They're associated with cancer. They're also associated with things like aging, because whereas things like proteins and lipids and other macromolecules in the cell, when they're damaged or broken down, they're generally replaced. So we talked about proteostasis in the cell. So proteins, as they're made, eventually they get broken down, but then they get replaced by more proteins. And that's true of lipids, polysaccharides. It's true of RNA even, but it's not true of DNA. The DNA in your cell generally does not get replaced until the cell is replicating. So if you have DNA damage, there are repair mechanisms for DNA, and we'll talk about those later. But once you have a DNA damage, it can be permanent or at least long term. And so aging is associated with a buildup, or can be linked to a buildup of these nucleotide transformations or DNA damage in a cell, which eventually leads to cell death, or at least leads to difficulty in repairing damaged tissue. Now, when you're studying DNA in the lab, you can take advantage of the way that DNA is synthesized to isolate it and to make copies of it. So if you want to study DNA, you can purify it from cells or from a tissue, just like you would any other molecule, like proteins, for example. But the only problem with studying DNA isolated or RNA isolated from a tissue sample is in any given cell, there's usually only one copy or two copies at most of any given DNA sequence. So you have usually just two copies of each chromosome in the cell. And so if you want a particular DNA sequence, you're not going to get as many copies of that from a given tissue sample, because there's only going to be one per cell, as you would of a given protein. So for a given protein, there may be hundreds of copies of a protein in a cell, versus, again, only one copy of a given DNA sequence. One way to study DNA sequences in the lab is to make copies of that sequence using a process called the polymerase chain reaction. So or PCR. So with PCR, you can make a copy of any given DNA sequence and amplify that region many times. So to do PCR, you combine basically four things. Your target DNA. So the target DNA is going to be the DNA that you've purified and isolated from your sample, and it's going to contain the sequence that you want to amplify. So maybe it's a certain gene, maybe it's just a certain region of a chromosome. It can be anything you want. That's the one nice thing about it. You may have heard of PCR recently, because PCR is used to one of the tests used to test for coronavirus. So coronavirus actually is an RNA virus. It carries around its RNA, but you can test for the presence of RNA just like you can with DNA by doing PCR. So your target DNA, in this case, would be actually based on an RNA sequence that you got from a patient that got a coronavirus test. So if you get a coronavirus test, that's usually what they're measuring, is they're looking for the presence of coronavirus RNA or DNA. It gets converted to DNA in your cells, but that's where the DNA would come from for the PCR. And then you add your deoxynucleotide triphosphate. So all of these are the four bases or the four nucleotides that you need to make a new DNA molecule. So when we say DATP, that means deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxythymidine triphosphate, deoxycytidine triphosphate. So those four bases are what you would need to make a copy of a given DNA sequence in their triphosphate form. So that means that they can be linked together. You need an enzyme that can combine DNA bases together to make a new DNA strand. And those enzymes are called DNA polymerases. So you add a DNA polymerase to your tube, which is going to actually carry out the process of making a copy of your target DNA. Now, in the old days, they would do this with just regular DNA polymerase. The problem is, and we'll talk about why this is in a second, but part of this reaction involves heating up your sample so that you can separate the strands. And the problem with that is most DNA polymerase enzymes are not heat stable, which means that, or thermostable, which means that they break down when you heat them up. So you have to use a special thermostable form of DNA polymerase. It's often called TAC. TAC stands for Thermus aquaticus. It refers to a species of bacteria that this enzyme is isolated from, that actually grows in hydrothermal vents down at the bottom of the ocean, where water temperatures can get well above boiling. So these are special versions of DNA polymerases that are stable at high temperatures. So you've got your target DNA, you have your nucleotide triphosphates, you have your thermostable DNA polymerase, and then you need primers. So a primer is just going to be a short segment of DNA, usually that you make synthetically. It's going to be an oligonucleotide, which means it's only going to be about maybe 10 to 15 bases long. And what's important is that the primers are going to be complementary to your target DNA, and they're going to bind to the target DNA at the 3' end of those strands. And the reason is that DNA polymerase will only add bases to the 3' end of an existing strand. So the primer is basically a marker that tells the DNA polymerase where to start copying from. So you mix all that together into a tube and stick the tube into a machine called a thermocycler, or just a PCR machine. And thermocycler is real simple. It's got this block of usually aluminum that has holes in it. Your tubes fit in those holes, and all it really does is it just heats up your sample to a very precise temperature for a very precise time, and that allows the reaction to happen in the tube. So a PCR reaction goes like this. You start with your target DNA, which again is always going to be double-stranded to start with. You heat your target DNA, and that will denature it. Again, DNA denatures at the amount of denaturation depends on the sequence, depends on the length. So you heat it up to nearly boiling to make sure that the two strands are completely separated no matter what they're made of. And then that separation lasts for usually just a few minutes, and then you cool it back down, not all the way back to room temperature. You cool it down to about 50 degrees Celsius, and that will be enough for your primers to bind. So not enough for the two target strands to completely reanneal, just enough for the primers to anneal. And again, the primers should bind to the 3' ends of your target sequence. So again, if this is your this represents your entire DNA sample that you've purified, but you only want to target a region within that. So this dark blue region is the actual part that you want to amplify. Then you choose primers that are going to bind to or be complementary to the 3' ends of each strand. So this is primer number 1, and it's going to bind to the 3' end of your target sequence. And this is primer number 2, which is going to bind to the other strand. So those are going to anneal to the template strand, and then you go to the extension phase, where actually you heat it back up, usually at about 75 degrees Celsius, and the enzyme will start copying. So it starts from the 3' end of each strand. This is the enzyme in green, and it starts copying towards the other end of both strands. Now, later on, we'll learn more about exactly how DNA replication works, but the important thing to understand is that it only goes one direction. So the DNA polymerase will only start at the 3' end of the primers. It will not go to the 5' end. And it'll just keep going until you heat the strand back up. So then you repeat step 1 again, basically heat the strands back up to 95 degrees. They separate again. But now you have your original target DNA, and now you have this new copy that you've made that only starts from where that primer was. And now you add you have more primers present, so there's way more primer than there is target DNA. So now the primers will bind again, but this time they will bind both to the original DNA and the DNA you just copied. And so those will copy also, and they will keep going. But this time, the DNA polymerase will stop once it reaches the end of that last strand that got made. And now the other one will go like it did before and just repeat what it did the first time. And so you'll end up with a bunch more of these longer sequences. But now what you have after this round, after again the DNA polymerase reaches the end, now you have this piece right here, this piece of DNA, which represents precisely your target region. So the part of the DNA strand that you were after now has been copied, and then this is the complementary sequence down here. So now you have one copy of that. Then you keep going. So you do that over and over and over again, and each time, again, that now you've copied that just that internal sequence, just that part you were after, and now after every cycle, this happens again. So you heat it again, the primers bind again, you extend the primers again. You end up with, every time you end up doubling the number of copies of your target sequence. And so each sequence takes about 10 minutes or so. And so you do this like 20 times. And after, 2 or 3 hours, you have many copies of your original target sequence. So it doubles every time. And so you can amplify your target sequence at least 1 million times. Then you purify it, and then you can do whatever you want with it. Then you can sequence it. You can use it for genetic modification, whatever you're trying to do. So the PCR is a very powerful tool. Again, let's say you're doing a coronavirus test. If a person had coronavirus, you would choose primers that are specific for a part of coronavirus genome. And so if you do PCR on someone who has the coronavirus in their sample, then you will amplify that DNA segment. Then you can look for that DNA on a gel or whatever you're using to visualize the DNA. Whereas if they don't, then the primers won't have anything to bind to, so the PCR, the reaction won't work. And so that person would test negative for the virus. So again, that's just one example. This is used in forensics sometimes. So if you have, you isolate DNA from a crime scene, for example, you can use this technique to amplify that DNA, even if there's just a tiny bit of it. So let's say it's just a little blood stain from a crime scene. Same way with paleontology or anthropology. If you have, let's say, a mummy and you want to purify DNA from that mummy, even if it's been there for 5,000 years, or from a woolly mammoth that's been buried in ice for 50,000 years. DNA. One thing about DNA is it's very stable chemically. So you can get DNA from very old samples. But of course, you can't get very much DNA that way. So PCR lets you make many millions of copies of a given sequence, and then study it without losing any, without having to worry about losing the original sample. Now, one of the things you might want to do with a DNA sample that you had purified by PCR is to sequence it, to find out what the sequence of bases is of the DNA sample that you have. Because of course, if you know the sequence of the DNA, then you can learn about perhaps what gene that DNA encodes for. Or maybe you can look for the presence of a mutation or whatever you need to do. And so there are lots of DNA sequencing methods that have been developed over the years. And in fact, and this is called the Sanger method. It's probably the most, or was the most commonly used and probably best known for a long time. And it uses what are called dideoxynucleotides. And a dideoxynucleotide is pretty simple. A deoxynucleotide is just a nucleotide where the ribose is missing an oxygen at the 2' carbon. A dideoxynucleotide is one where the ribose is also missing an oxygen at the 3' position. So to do dideoxy sequencing or Sanger sequencing, you take your template strand, which is the DNA you're trying to sequence. So this is the template strand. Again, you have to start with a primer. If you're going to make a copy of DNA, you can't just have a template strand. You have to have the 3' end of an existing strand to start from. So that's your primer. So you have to have something to start off at. And then you add your, again, your DNTPs, your regular nucleotide triphosphates, and one of the 4 bases in its dideoxy form. So here what they've done is they've added the primer, the template, the 4 deoxynucleotide triphosphates, and in this case, dideoxy ATP. So dideoxy ATP, or the other 4 bases will get added along. So if your DNA polymerase is going along, again, starting at the 3' end of the primer strand, if it sees a C in the template strand, it will add, it will grab a molecule of deoxy GTP and add it to the 3' end. It will form a phosphodiester bridge between the C and the G. And then if it gets to a T, it will try to grab a deoxy ATP. So if it finds a deoxy ATP, it will add it, and then it'll just keep going. But if dideoxy ATP is present, it will grab it. So the enzyme doesn't really care what the base is. All it cares is that it hydrogen bonds with the base on the template strand. So the A will hydrogen bond with the T on the template strand. And because there's the triphosphate group is attached to the 5' end of that dideoxy nucleotide, it will form a bond, a phosphodiester bond between the 3' end of the last bond it created and the 5' end of the dideoxy. But because now this dideoxy nucleotide is missing an oxygen at this 3' position, there's nothing for the enzyme to add to. So it will essentially stop when it gets there. So it'll get to that A and it will just quit. So to use this for sequencing, you set up, or at least the old way to do it was to set up 4 different reactions. One reaction, or all 4 reactions would contain your template DNA, would contain your template, and would contain your 4 regular deoxy nucleotide triphosphates, so DATP, DCTP, DGTP, and DTTP. But then into each reaction, you would include one of the dideoxy forms of each of the nucleotides. So one reaction would include dideoxy ATP, one would include dideoxy CTP, a different one has dideoxy GTP, and then dideoxy TTP. And then in each case, you would then end up with a bunch of pieces of DNA, single-stranded DNA, which would start with your primer, and usually the primer is labeled. So in the old days, they would label it with a radioactive isotope, maybe your isotope of phosphorus or carbon or hydrogen, whatever one they wanted to use. That just lets you see it. And then the other end would include a dideoxy nucleotide. So in the tube that contained dideoxy ATP, you'd have a bunch of pieces of DNA, which would have a radioactive primer at one end, and then a dideoxy A at the other. And the position of that A, or the length of that piece, would correspond to how many bases there were between an adenine and the primer. So because again, if the enzyme, the DNA polymerase, saw a T in the template strand, it would add either an adenine, a regular adenine, or a dideoxy adenosine to the template strand. If it added a regular adenine, it would just keep going. So this here and here, for example, represent pieces where it saw the T in the template strand. It happened to grab a regular adenosine instead of the dideoxy adenosine and just kept going. But here represents a case where it saw that T and grabbed a dideoxy adenosine instead, and then it stopped. It couldn't keep going after that, so it made a very short sequence. And then in this complete sequence contained had 3 thymines on the template strand, and so should have had 3 adenosines, but in each case a dideoxy adenosine got added at a different location. So you have 3 different pieces of different lengths. Each length tells you where that A is in relationship to the primer, and each reaction has a different combination of pieces, and each one has pieces of different length that correspond to that base. So you take each of those reactions after they're done, run them on an electrophoresis gel. You can do electrophoresis with DNA just like you can with proteins. So they run in a gel. Based on length, the shorter pieces go farther than longer pieces. And because you have pieces that are whose length corresponds to the position of those bases, you can read this graph and figure out the sequence. So the shortest piece will be the piece that has a base closest to the primer. And so it happens to be in the tube that contained dideoxy-GTP. So what that means is that this band right here, the shortest, the one that moved the farthest, therefore it's the shortest, it corresponds to a G at that position closest to wherever the primer was, so toward the 5' end of the complementary strand. So that band tells you that there's a G at this position. And then you look and see what's the next one. The next shortest one came from this lane, which contained dideoxy-ATP. So this band means that there was an A at this position. And then the next two bands are both in the lane that contain dideoxy-TTP. So that means you have two T's in a row. And you just go all the way up the strand. And you can a typical audio radiogram like this would be, could be many hundreds of bases long. Now, more recently, and then in fact, it's really the case that this method is not used as much as it used to be because obviously it's very labor intensive. You have to do 4 separate reactions, and you have to have a gel. If you want to sequence a region that's more than a few hundred base pairs long, you have to do this many times in order to sequence. If you wanted to sequence like an entire chromosome, which is millions of bases, you would have to do many of these reactions. There are more modern ways to do it that instead of using radioactively labeled primers, you use fluorescently labeled dideoxynucleotides. So in this case, instead of labeling the primers, you label the nucleotides, and you use fluorescent tags instead of radioactive tags because the fluorescent tags, you can use different colors. Fluorescent just means that when they emit light of one wavelength when they're exposed to light of a different wavelength. And so there are lots of different fluorescent dyes you can use in this way. And if you combine those with the dideoxynucleotides, you can create fluorescently labeled pieces of DNA that again are going to have lengths that correspond to their position. So you do the same exact reaction and again run these through a gel, but this time all of the dideoxynucleotides are all in the same reaction tube. You don't have to have 4 separate reactions. You can put them all in a gel, run them through the gel, and then at the end there's a detector which shines again a light through the gel. Each base or each fluorescent tag will fluoresce at a different color that you detect with a detector, and then that can be attached to a printer, computer, which then creates a printout like this, basically a graph that corresponds to the intensity that the detector detected for each different wavelength that can be fluoresced. And so each peak, therefore, corresponds to a different base. So if your cytosine is labeled with a blue fluorescent tag, then every peak on in the blue part of the spectrum corresponds to a C. If your thymidine is labeled with a red fluorescent tag, every peak in the red part of the graph represents a T. And so you can just look at the graph. They basically just printed all 4 graphs at once, and then color coded them. So then you can just use this for determining the sequence. In fact, nowadays you don't even get a printout. You just the detector or the computer just compiles the sequence data into usually just a big long text file, and then you just download that to your computer, and you can then use it. Use computer algorithms to look for particular sequences. In fact, nowadays this method is almost entirely automated. So you load your sample DNA into a machine. The machine already has all the components for the reaction present, and it just does all the steps for you, and then just gives you a sequence at the other end. In fact, in most labs, modern labs that do at least large amounts of DNA sequencing don't even really do the sequencing themselves. A lot of times you can send your DNA sample off to, if you're at a big university, your university like OU has a DNA sequencing core facility. So everybody that needs DNA sequencing done just sends it to this one lab to do the sequencing, or there are companies and labs that specifically do sequencing for you. This is how whole genome sequencing involves lots of different would involve a lot of work if you were to if we were trying to just do it one piece at a time. So there are a lot more sophisticated techniques that involve complicated machinery to and computational algorithms to determine DNA sequence. But it's still useful to understand how DNA sequencing was done before modern technology made it easier. Okay, the last thing I'd like to say about nucleotides is, of course, we know that DNA is important for storing genetic information, and later on we'll talk about again how it's replicated, how DNA sequences are transcribed into RNA, how RNA is translated into protein. But there are other things that nucleotides do apart from just store genetic information. One important one is in the storage of energy. So nucleotide triphosphates are important energy storage molecules for most organisms. Of course, when we say triphosphate, what we mean is a nucleotide with 3 phosphates instead of just 1. So an NTP means nucleotide triphosphate, NDP is nucleotide diphosphate, NMP would be nucleotide monophosphate. So the phosphate that's directly attached to the 5' carbon we call the alpha phosphate, and then the next one is the beta, and then the gamma. And so all the nucleotides have monophosphate, diphosphate, and triphosphate forms. So when we talk about ATP, what we mean is adenosine triphosphate. So that's the triphosphate form of the ribonucleotide adenosine. And then if you're talking about deoxy nucleotides, then you have DAMP, DADP, and DATP, and so on. Now, the reason that these are useful for storing energy is because the anhydride bonds between the phosphates, that's what connects the phosphates to each other, have a lot of energy available when they're broken, compared to the ester bond between the phosphate and the 5' carbon. So there's about 30 kilojoules per mole for each of those bonds. So breaking those bonds is a good way to extract energy. That's why an ATP is typically how energy is stored in most cells, although GTP is also used sometimes. But producing ATP from AMP involves a lot of energy input, but then there are lots of enzymes that can break that anhydride bond between the alpha and beta phosphates and extract that energy back out. So and we'll talk about how ATP is made and how it's used more later on. Nucleotides can sometimes be used as cofactors for enzymes. So a cofactor is a molecule that an enzyme uses as part of a enzymatic reaction. And so, for example, coenzyme A is a modified form of adenosine. It's basically adenosine with this sort of long hydrocarbon chain attached to it. And it's involved in a lot of reactions, especially a lot of transfer reactions. So this is an example. So this enzyme, choline acetyltransferase, is responsible for making this neurotransmitter here called acetylcholine. And acetylcholine is made from choline. This is the choline group, and by adding an acetyl group. And so the enzyme choline acetyltransferase adds an acetate group to choline, but it uses this coenzyme A as kind of a carrier group. So it grabs acetyl-CoA, breaks off the acetyl group, adds it to choline to form acetylcholine. So it's just an example of how a nucleotide can act as a cofactor, and there's lots of other cases where this things like coenzyme A show up. These are two really important enzyme cofactors: nicotinamide adenine dinucleotide. So it's basically an adenine or adenosine group with another nucleotide-like structure called nicotinamide that's linked by two phosphate groups. So this is called NAD plus, and this and then another one called flavin adenine dinucleotide. Again, you have an adenine linked to another nucleotide-like structure called riboflavin. That's called FAD. Both of these are important for oxidation reduction reactions. So in a lot of various, these act as electron carriers. So NAD plus is the oxidized form, NADH would be the reduced form, and then FAD can be reduced to FADH2. And in both cases, by picking up those electrons, they can act as carriers of energy, and so they're important for the reactions involved in converting chemical energy from food into producing energy storing molecules like ATP. So we'll come back and we'll hear about NAD and FAD a lot more later. And I already mentioned a little bit that there are versions of these nucleotides that can act as second messengers. So cyclic AMP, this is cyclic AMP, where actually in this case it's a 3' 5' cyclic monophosphate. So the phosphates connect to the 3' and the 5' carbons of adenosine. And then cyclic GMP is basically the same structure but with guanine instead of adenine. And these act as second messengers. And so what that means is these are produced as part of complex intracellular signaling pathways. So there are enzymes. So for cyclic AMP, for example, it's produced by an enzyme called adenylyl cyclase. Adenylyl cyclase is activated by proteins called G proteins. Those G proteins in turn are activated by certain types of receptors on the cell membrane, which in turn are activated by extracellular signals like certain neurotransmitters or certain hormones. And so these second messengers then can go on to activate other downstream targets, things like kinases, certain transcription factors. So a whole network of molecular signals can be activated, kind of propagate through the cell using these kinds of molecules. So these are just some examples of how nucleotides function in the cell apart from being part of DNA. And that is the end of that chapter. So, but we will in the next chapter talk about some of the things you can actually do with DNA.